Okay, so we said we'd start with the nucleotide as the next part of looking at DNA. Okay, so nucleotides are the monomer units of DNA. So essentially we could call it a DNA monomer unit. All right. That means it's just a single unit that repeats over and over and over and over again to make up DNA, which is considered a polymer, which is made up of many of these repeating units. So we want to know, what is a nucleotide? Well, the nucleotide is made up of 3 parts. Okay, so there's 3 parts to a nucleotide. There is a 5-carbon sugar. There is a phosphate functional group. And then there's something called a nitrogenous base. Okay. Now, we're going to start off with and focus on the structure of the sugar, okay? This 5-carbon sugar. Because it's sort of what kind of sets up one of the differences between DNA and RNA. The sugar, in a way, is kind of the core of the nucleotide structure. The sugar sets up things that we talk about with DNA and DNA replication, terminology, where we use 3' and 5'. That also relates to the sugar. So if you can draw the sugar on your own just from scratch, that's going to help you really picture not just what's a nucleotide, but how the whole structure of DNA even fits together. All right. So in drawing this 5-carbon sugar, we're going to draw it as a ring, not as a linear structure, because that's how it exists. So we're going to start off with an oxygen, right, like that. So that's oxygen. Now that oxygen is going to be attached to a carbon. Okay. 2nd carbon here, 3rd carbon here, 4th carbon here, and then these carbons are joined together. Okay, so it forms this ring structure. Right? Sometimes it's drawn like this. I'll draw it maybe down low, kind of draw the same thing. The O, but then these are just drawn like that. Okay, so that's the oxygen here. And then we don't draw the carbons typically. When we draw the ring, each corner, each little angle we know is representing a carbon. So for now, I'm going to put them up here, but in the future, I won't, and you have to remember that there. But I said it's a 5-carbon sugar, right? So there's carbon number 1, there's carbon number 2, there's carbon number 3, and number 4. But that's not carbon, that's oxygen. So carbon number 5 is actually stuck out of the ring. Okay, so it sticks up like this. Now, for this structure, ribose, following the structure of a sugar, okay, sugars are supposed to have oxygen. Sorry, every carbon of a sugar is supposed to have an oxygen, right? So if it's C5, it should be O5, okay, 5 oxygens. And so we're going to have oxygens as OH groups. Stuck to these. Now this carbon, that's its oxygen, and then there's going to be an OH here. I'm not drawing them exactly, right, but you can see that. Now the carbons are supposed to have 4 bonds, so then there would also be hydrogens. Okay, so this one here would have a hydrogen. This one here would also have a hydrogen and a hydrogen. That would be 3. Then this one just has a single hydrogen. That would be 1, 2, and this one would have 2 hydrogens, like that. All right, now that's the 5-carbon sugar. That sugar that we just drew here is called ribose. You should be able to draw this sugar. Right? So another version of it, would be simply like this, showing OH, 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 and then there, because that's that, and then another OH up here. Okay, so there's another version. Make it look more like an H, okay? All right, you got that? So whole carbon number, different color, so we just know what we're talking about. Carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5 distributed like that. 1, 2, 3, 4, 5. Each one has the oxygen. This oxygen has two bonds like it's supposed to. Remember, in the sugar, there's usually a carbon-oxygen double bond, but in the ring structures, they're kind of split apart. The oxygen is inside that ring. Now, I mentioned DNA before being copied into RNA. So if we want to think about or talk about for just a moment, what's one of the differences between DNA and RNA? And obviously you can be really simplistic about it and say, oh, this one has a D and that one has an R. And you're not totally off, on that oversimplified explanation. So we could ask, what does they mean? Well, what is a D and what's an R? What does that mean? So RNA. Well, RNA stands for ribonucleic acid. Okay, so ribonucleic. And then DNA stands for deoxyribonucleic acid. Okay, so RNA, and then there's the DNA. Now here I just said this is the sugar ribose. So what we're going to find is that this is the sugar that we find in RNA. That's what it looks like. And now if you want to see the difference, what does the sugar look like in DNA? Is the sugar the same or different? Well, it's a different sugar. It's called deoxyribose instead of ribose. So if this is ribose, and we want to draw the other sugar, we want to draw the DNA sugar, so we have to draw deoxyribose. What is it going to look like? Watch this. Now it's deoxyribose. Okay. The ribose sugar found in RNA and the deoxyribose sugar found in DNA are almost exactly the same sugar. The difference is here at the number 2 carbon. In ribose, I can't block it from you unless I kind of go on the other side of this screen here, but in ribose, you have the OH there, where it's supposed to be. It's typical sugar. Okay. And then deoxyribose, we get rid of that oxygen. It has one less oxygen. It's deoxy. So it kind of breaks the rule there for the sugar, but that's just its structure. All right. So right now you should be able to draw, or with practice, you have the information you need to draw the ribose sugar, which is the core part of a nucleotide, right? The 5-carbon sugar. And then you can modify it to be deoxyribose. So if I said draw the DNA sugar, the RNA sugar, it doesn't really matter. It's pretty much the same sugar. You just have to know about the oxygen at the number 2 carbon. Okay? So that's the first part. The second part is the phosphate functional group. Okay? What's a phosphate functional group? You may remember this, or you may not. So sometimes it's written as PO4 -2, right, like this. So it's phosphorus in the middle, surrounded by, say, O4. So 4 oxygens. 1, 2, 3, 4. Now, what else are we going to have here? This phosphorus is going to have a double bond to one of those oxygens. So there's actually 5 total bonds here. One of these oxygens is going to be bonded to essentially this carbon right here. Okay, so there's going to be a bond. So I'll draw this. This is the bond. This is going to go to the phosphorus here, and that's going to be I'll do the carbon in the pink so we can kind of keep the color consistent. Is going to be a carbon. Now that's not part of the phosphate functional group. That's a carbon from the sugar. Okay, here, but that's where it's going to go. The other thing you need to know is the -2. So the other 2 oxygens are going to have a negative charge. So the phosphate is negatively charged. It's always going to be negatively charged. It'll carry this -2 charge. And it's going to be always attached to the number 5 carbon. Okay? So you have the sugar. Then you have the phosphate. And the phosphate's location is always on the number 5 carbon. Okay? Last part is the nitrogenous base. So I'll draw those in a separate video. We could look at their structures. Typically in an intro general biology class, you wouldn't be having to draw then those as well. Okay, so but you need to know what they look like, and you need to be able to identify them if given to you. So we'll put those into the next video. Okay. What we want to know, and what I want you to be able to show is, okay, so where would the phosphate functional group go? Okay, here. All right, so now we have phosphorus attached to the number 5 carbon. To the number 1 carbon, we're going to put the nitrogenous base. Okay, so that's where it's going to go. What is the nitrogenous base? Well, for now, we're just going to simplify them as things you probably have heard of. The A's, the T's, the G's, and the C's. So adenine, thymine, guanine, cytosine. Like I said, that will be the next bit of information we'll get into. Is after we show how to put these together a little bit more, then we'll go into some detail on the A's, T's, G's, and C's when we're going to put 2 strands together. Right now, we're just building the 1 strand, and for the single strand, this is kind of like a unique tag sticking off it. Does it have an A? Does it have a T? Does it have a G? Which one does it have? Okay. Later, those structures will become important. If you're going to take 2 strands of DNA, what's going to happen is, I'm kind of jumping ahead a little, but get it here. There's going to be a sugar molecule, and then it's going to be attached to a phosphate, attached to a sugar, attached to a phosphate, attached to a sugar, attached to a phosphate, attached to a sugar, attached to a and so on. There's going to be a chain of phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar that's made, and it's going to be exactly the same and repeat over and over and over and over and over again. Nothing unique or different about it. That's going to be a strand of DNA when we get to that point. It's just the next step. What's going to be unique about it will be what's attached to the sugar will be a certain nitrogenous base. So maybe this one is A, this one is T, this one is C, this one is G, for example, because they could all be A, they could all be G, but you get the idea. The sugar then has a certain specific nitrogenous base attached to it, and that'll make this nucleotide unique. There's really only going to be the 4 nucleotides in DNA, the ones carrying the A's, T's, G's, or the C's. And then in RNA, which we'll get to, T is substituted, T is replaced by U or uracil. Okay. In general, there's some other things that go on there too. So right now, there's one last little thing to add into us. The number 1 carbon has the nitrogenous base. In ribose, the number 2 carbon has an OH, the number 3 carbon has an OH. The number 4 carbon is just stuck part in here, there's nothing special attached to it. And then number 5 carbon has the phosphate. Now, in DNA, because we're going to be focused on DNA here, so the deoxyribose, missing that oxygen there. Which also means we don't have the OH group anymore. It's not there. When we start to build this chain, this sugar, phosphate chain, there's going to be 2 parts of this that are then going to interact with other nucleotides. So say if I took this nucleotide here and I joined it to the one I started to draw down there, right, and I wanted to put them together, the bond is going to be between the OH, the number 3 OH, and the number 5 carbon phosphate, a 3 to 5. Okay. This guy's not going to be involved in any of these bonds. Okay, so the number 2 one isn't really going to be involved in doing anything else. The phosphates will on the number 5. The OHs, the hydroxyl groups, will on the number 3. And the nitrogenous bases will later, when we draw a second strand next to this one and have to join the 2 together, then that's when they'll come into play. But the number 2 one isn't going to do anything. And the number 4 one, they're involved in the structure of the sugar, but they're not involved in making other bonds with other either parts, other nucleotides, or anything else. Okay. So the 3 and the 5 are the ones that we're going to talk about most to become important. But we usually aren't going to just say 3 and 5. We're going to say 3 prime or 5 prime. You might ask, well, why is he going to say that? Well, like I said, I didn't draw the structure of the nitrogenous base yet. We'll be doing that next. But we're going to find nitrogenous bases are ring-like structures, but they're going to alternate carbon, nitrogen, and so forth. So the thing is, they're not just made of nitrogens, they're made of carbons and nitrogens. So if we talk about a nucleotide as a whole structure, and we want to talk about a specific carbon, we say, oh, the number 1 carbon. Someone may not know if you're talking about the carbon of a nitrogen or the carbon of the sugar. So we call all the carbons of the sugars by number with a little prime. So 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. That's just sort of the numbering of them. Okay. Now, we're ready to put them together. Okay. Kind of get at that idea. First, we're going to just show where it happens, and then we're going to pose a problem for it to actually happen. Okay, so I'm going to redraw. I'm going to draw another one here, DNA nucleotide. A lot of times we just draw it like this. Okay, N. All right, so really simple. Add to it, really just for clarity, 1, 2, 3 prime, 4 prime, 5 prime. We know that has the five carbons. Now what we're going to get is a bond, ultimately, between this phosphate and that OH. That's how the 2 nucleotides are going to be joined together. Or essentially, I should say that's where they're going to be joined together, the location. So then it's phosphate, sugar, phosphate, sugar, and then like I said over here, it's going to repeat just in a pattern. Well, it'll just happen 100 times, thousands times, 10,000 times. You know, you only have to see it once, really, or twice to see what's going to happen, and then it's just going to repeat itself over and over again. The problem here that I want to get at before we move on to something new, totally new, is energy. We're creating a new bond here, and that's going to make this a more organized structure. Things don't organize unless energy is invested. We don't see where the energy comes from right now. We're sort of like, I don't know, you know, maybe. And so one thing people can say is maybe it comes from ATP. And you would not be totally wrong, but it's probably not in the way you think, unless you already know the structure of nucleotides. And if you do, you'll realize ATP is a nucleotide. All right, so the structure of an ATP molecule, right, is going to be, I'm going to draw this really small here, the A nucleotide. This is, if you recognize, that's a ribose sugar, TP, that's triphosphate, right? 1, 2, 3 phosphates, okay? That's the structure of ATP, but you're saying, wait, that's what we just drew. Nitrogenous base, of which one of those could be a sugar. In this case, it's the ribose sugar. Notice I have the O there, right? It's not the DNA one. And then it's 3 phosphates, though not 1. But it's the same. It's not essentially the same thing. It is the same thing. It's the same thing. So if you think about that a second, you say ATP will, you know, TTP. Really, we could substitute this. So A, T, right? G, that's GTP. So we essentially have ATP, TTP, CTP. Oop, forgot that. Messed that one up. And GTP. Okay, so the A, T, G, and C of DNA exist in triphosphate forms. So what's that mean? Essentially, I was just talking about energy, right? I said this is going to need energy. Well, before the bond forms, before it forms the nucleotide, the thing that we just talked about, it actually had 3 phosphates. They all have 3 phosphates when they're just sitting alone by themselves. When this bond is made, an enzyme is going to break off 2 of them. The diphosphate is going to be broken off and energy released. When that energy is released, it can create this new bond, okay? The bond that was called the phosphodiester bond. All right, this one here. So if you need the spelling of it, phosphodiester bond, that's that bond right there. That's the bond between all the nucleotides. And the energy to make that new bond is going to come from the individual nucleotides themselves. So they're going to bring their own energy essentially with them because they are ATPs and to GTPs and TTPs and CTPs. They are molecules carrying 3 phosphates that can have some of them broken off and then release energy, and they can be used to then make bonds to join them all together in long chains. So DNA itself is a polymer. It's made up of smaller subunits called monomers, which in this case are nucleotides. When we look at a DNA structure, there's only going to be a single phosphate, sugar 1 phosphate, and so on. But before those monomers are assembled, they're actually going to have 3 phosphates, right? And then they're going to use the energy from those other phosphates to make the bond. And so we could just repeat this, like I said, 3 times, 5 times, 100 times, 10,000 times, and it would just be the same thing over and over again. A new nucleotide with 3 phosphates come in, 2 of them are broken off, the new phosphodiester bond is formed, repeat, 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 and you get a strand, a single strand of DNA. Now DNA is going to be a double-stranded molecule, and so that's the next topic.